Hello, my name is Dr. Erin Young, and I'm a bioinformatician at the Utah Public Health Laboratory. And today I'm going to be focused on some nanopore sequencing that we've done of some antimicrobial resistance organisms. I do have a disclaimer. Please don't hold anybody accountable for what I say today other than myself. Whole genome sequencing is used in public health to better understand what is making us sick and why it is evading therapeutics and other treatments. There are a bunch of different programs at the CDC, and so I stole this slide here from the CDC, or this webpage from the CDC, but there's foodborne outbreak investigations, antibiotic resistance, and there's a lot of other capacity for sequencing of bacterial isolates. For those not in public health, I just want to let everybody know that I'm not CDC. I am at a small lab a lab here in Utah, and I am going to be talking about antimicrobial resistant organisms. So for the ARLN lab network program from the CDC, the United States is divided into a variety of sections, and Utah is the main lab for the AR lab network in this region of the United States, the blue region. There's also a similar structure for other organisms such as PulseNet, which we've also done long-read sequencing for. So when I talk about the organisms that we've sequenced for antibiotic resistance, most of them have come from the region of the United States, although we do have some isolates that were shared with us from other regions. So for the ARLN lab network program, this is the general whole genome sequencing process, where it starts with a bacterial culture, and bacteria are isolated into colonies, and in theory that means we're doing like some sort of pseudo single cell sequencing without having to deal with the single cell prep in bioinformatics, where we take a single colony, which is supposed to represent like an original bacteria that just replicated multiple times, that is sequenced or isolated, and then prepared with library preparation kits, and then sequenced. This is a MySeq. Most of the ARLN isolates are sequenced with Illumina sequencing. And then we get this cool data, which looks exactly like this. This here's, I know it's a little small, but it says reconstructed genome. And, you know, to an extent, it is a reconstructed genome. Most of what our assemblies are de novo assemblies. And from this, we can determine the antibiotic resistance genes, serotypes, and other genetic information from this information. But rumor has it, and I'm going to keep spreading this rumor, that if the Illumina sequencing with their kind of incomplete genome assemblies are going to give us information, Nanopore has longer reads and can potentially complete the genome to a higher degree than that of Illumina sequencing. But first, we have to make sure that it's actually doing everything that Illumina is doing, which I've made this kind of non-exhaustive list of benefits of Nanopore. If we're going to use Nanopore sequencing for antibiotic resistance, public health surveillance, and other things, we need to be able to identify AMR genes. We need to be able to serotype or subtype our bacterial isolates. We need to be able to have high quality assemblies that we can use to compare to other high-quality assemblies to see if bacterial isolates are linked in some sort of outbreak. But I think we can do more with nanopore sequencing, such as identify where those AMR genes are. Are they going to be chromosomally located? Are they going to be on a plasmid? Plasmids are more likely to be transferred between one bacterial organism to another. And so if you're trying to contain some sort of antimicrobial resistance gene, being able to track where it is in the organism is significant. Additionally, nanopore sequencing has the potential for a faster turnaround time. And for reservations, there's a couple of reservations of using nanopore sequencing, but the one I've listed is just a decrease in quality, because that's generally what I hear people are concerned about. And so this is what we've sequenced at UPHL. We've sequenced a variety of organisms. Most of these are antimicrobial resistant. Things that I reflux to long-read sequencing are those that have an incomplete AMR gene, or those that have a oxygen that hasn't been classified, or other AMR gene that hasn't been classified, or if an suspected organism is part of an outbreak. So these are not necessarily surveillance activities. There is a bit of a bias into what organisms are long-read sequenced. But we do have a variety of different organisms that we've sequenced. So far, it seems like if you have a bacterial isolate and you can extract the DNA, you can probably nanopore sequence it. Also, we do have a few years of experience with nanopore sequencing. We started some nanopore sequencing back in 2019, and then we had a stretch where our energies were focused elsewhere, and now we're back to developing our nanopore sequencing and identifying its utility. Antibiotic resistance is bad. It is expensive, and it decreases life expectancy. There are ways that antibiotic resistance happens and that it spreads, and these public surveillance programs help us identify what is causing antibiotic resistance and what organisms it is and how it's spreading. For the rest of the talk, I'm actually going to be restricting my results to the last 76 isolates that we sequenced because they're with the R10 flow cells and the most recent chemistries. And I don't want to be like a broken record talking about how the quality has improved substantially. Basically, in summary, we looked at MLST types in these isolates, and they were the same between Illumina and Nanopore. We looked at the predicted genus and species, and they were the same. Plasmid predictions were a little bit different from Plasmid Finder, in that Illumina, we do see occasionally that plasmids are predicted more often in Illumina data than in assembled Nanopore data. And just restricting further to just the isolates that carry beta-lactamases. The beta-lactamases are a family of AMR genes. We had 59 isolates that carried at least one beta-lactamase gene, and 42 out of these 59 had exactly the same OXA profile between the Illumina sequencing and the Nanopore sequencing. There could be like laboratory and isolation processes that could account for differences, which I'm not going to really cover. I'm just covering kind of what we saw. In one of these 59, we noticed that there were oxygenes called in the Illumina sequencing, but not in the Nanopore sequencing. And we didn't identify any SNPs in these beta-lactamase genes. that we were looking at between the Illumina and Nanopore data. When we looked at the Nanopore data, though, we did notice that we were more likely to identify beta-lactamase genes in the Nanopore data. Again, these 59 isolates aren't necessarily unique. Some of them are part of outbreaks. So there were 7 that were all predicted to be part of the same exposure and part of an outbreak. And in all 7 of these, they were shown to carry OXA10 in the Nanopore sequencing, but none of them had OXA10 in the Illumina sequencing. And we have observed this as well for OXA23 family member in an additional isolate. In several of the isolates, we observed a duplication of the AMR genes, which for a lot of the Illumina sequencing, it's really just presence or absence. But in the Nanopore sequencing, we can see multiple copies of things like OXA2 or OXA23. And with that, I want to thank everybody at the lab. I'm a bioinformatician, so everybody who did the laboratory magic is here on this slide, and I'm very grateful for them. Also, this couldn't have been possible without an AMD pilot project grant. Thank you for your time.